All right, let's jump right in. When you think about the active compounds in cannabis, your mind probably goes straight to THC or maybe CBD, right? They get all the attention. But there are over 100 of these compounds, and today we're pulling back the curtain on one of the lesser known ones, cannabichromine or CBC for short. And that's really the big question we're tackling today. What actually happens when CBC gets inside of us? It turns out the real story isn't about what CBC is, it's about what our body turns it into. And that transformation is a fascinating piece of biochemical detective work.
So let's start with our main character here. What do we actually know about cannabichromine? Well, scientists have seen its potential for a while now, but the full story, that's been a bit of a mystery.
Now, what's really interesting is why it's non-psychoactive. You see, unlike THC, which famously latches onto the CBR1 receptor in your brain to produce that high, CBC, well, it doesn't really do that. Instead, it seems to play with other pathways in the body tied to pain and inflammation, which is exactly why researchers are so interested. But, having potential is one thing. Knowing how it actually works is a whole different ballgame.
And this right here, this quote from the research paper we're digging into today, it just hits the nail on the head. We know CBC gets into the body and it seems to do something, but the how, the whole process of it being broken down and transformed, that's been a total black box. Well, until now. So where does all this magic happen? Our investigation takes us to the body's #1 chemical processing plant, the liver. This is where the real mystery of CBC's transformation begins to unfold. Seriously, think of your liver as this incredibly sophisticated biochemical laboratory. When a compound like CBC arrives, it doesn't just pass through. Oh no, it gets taken apart, analyzed, and completely re-engineered by a team of specialist enzymes. They might add an oxygen atom over here, or break a bond over there, totally changing its structure and its function. And these are our lab technicians, the key players in this story, the cytochrome P450 enzymes, or CYPs for short. These things are absolute workhorses. I mean, they're responsible for metabolizing something like half of all prescribed drugs. They are the body's ultimate transformation artists. But here's the thing. These master chemists, they can't just work in the dark, right? They need energy to pull off all their chemical reactions. And that power is supplied by a supercritical partner enzyme called CPR. You can think of it like a battery pack, giving the CYP enzymes the juice they need to get the job done. Okay, so we have the location, the liver, and we have our suspects, the CYP enzymes. So the next step for the researchers was to track exactly what these enzymes were turning CBC into. What were the results of all their hard work? And here's the first big clue they found. It turns out that when the CYP enzymes got to work on CBC, they created two main new molecules, or what we call metabolites. Let's just call the one from the top pathway the hydroxy metabolite, and the one from the bottom pathway the epoxy metabolite. CBC was essentially putting on new chemical disguises. Now, this was a huge step, but it immediately brought up the next question. You see, there isn't just one CYP enzyme. There are dozens of them. So which specific one was doing all the heavy lifting? Who was the most efficient CBC transformer in the liver's entire lab? And the answer was stunningly clear. While a few different CYP enzymes could process CBC, one was an absolute superstar. This chart shows the rate of production, and as you can see, an enzyme called CYP2C9 was dramatically more efficient at metabolizing CBC than any of its colleagues. It was our prime suspect for sure. But here's where the investigation took a really cool twist. What happens in a controlled lab setting isn't always what happens in the messy, complex environment of a living body. When the researchers gave CBC to mice, they found that even though the enzymes can produce a mix of metabolites, in a living system, one of them just took over. That hydroxy metabolite we saw earlier became the dominant one found in the blood. And the plot just thickened even more. This dominant hydroxy metabolite, it was like a ghost. Its levels in the blood peaked just 30 minutes after it was given, and by the 2-hour mark, it had completely vanished. It was there, and then it was gone. This suggested the story was far from over. And that leads us to the most incredible discovery of this entire investigation. These metabolites, these new molecules our body creates from CBC, they aren't just byproducts waiting to be thrown out. No, they have secret powers of their own. Okay, this is the climax of our story right here.
It turns out the body's transformation process creates a cocktail of compounds that have opposite effects. Some of the metabolites, like the ones you see here on the left, are powerfully anti-inflammatory. But the major metabolite they found in the mice, the ghost, that hydroxy one, was actually found to be pro-inflammatory. It actually promoted inflammation.
And the anti-inflammatory effects weren't just a little thing. The research showed that some of these other metabolites could reduce key inflammatory markers by nearly half, 50%. So our bodies are creating both a fire starter and a fire extinguisher from the exact same initial compound.
This discovery completely reframes how we have to think about CBC and maybe a lot of other compounds too. The true therapeutic effect, or even the potential side effects, might not come from the drug we take, but from the unique, personalized blend of active metabolites our own body chemistry creates.
So, after this deep dive into the liver's lab, what's the big picture? What are the key takeaways from this whole biochemical detective story?
Well, first, a compound's real power might actually be in its metabolites. Second, this process can be a double-edged sword, creating substances with opposite effects. Third, understanding this is absolutely critical if we want to design better, safer therapies. And finally, and this one is kind of personal, your unique enzyme makeup, like your levels of that superstar CYP2C9 enzyme, could be the reason you respond differently to something than your friend does. And that leaves us with a final, really provocative thought. We spend so much time and money searching for the perfect molecule to use as a drug. But what if the real pharmacy, the real source of healing, isn't in a pill at all, but is already inside us, just waiting for the right ingredients to start its own secret chemistry?